Okay, yeah. So thanks a lot. And yeah, well, we'll try to replace Max, but that's going to be hard. So today I'll tell you a bit about the work that we've been doing here at TPFL, and particularly work that we started around 2 or 3 years ago in terms of exploring. Basically, the way that I like to think about this is to give a new perspective into protein molecules. And so I know that many of you don't really think about proteins on your daily basis. So let me just kick off with 2 slides of introduction, because, you know, in our lab, all we think about is proteins on a daily basis. So, but, you know, just to summarize it for you, ultimately proteins are the workhorses of living cells. They're also the target of most of the drugs that you actually take. And the way that this works, it actually resembles a lot what computer science is based on, which is basically memory storage and codes. And it kind of works a little bit like a code, right, where you have genetic information that is encoded on your DNA, and then this genetic information is translated into polymer sequences of strings of amino acids, which importantly, and this is a big field of study in biology, then adopt a 3-dimensional structure, which is responsible for function. And so thinking about many of the functions that these proteins can do, I'll present you a first one, right? So all of you are here because hopefully you got vaccinated and now you have antibodies against COVID. So here you go. Those are some of the most essential proteins that are done in our body. But it really, anything you can think about. Protein sequences can encode and can have functions in living cells like communication, catalysis, structure. Okay, so but in the field of studying protein biochemistry, one of the things that we tend to do is we need to represent these molecules in some way. And so for this audience, also what I don't need to tell you is that data representation in any domain is key. So in proteins, it's kind of the same deal, right? And the thing here, I'm showing you actually multiple ways of representing the same object. And you can, for instance, think about these molecules as just its architectural components, which are basically composed of alpha helices and beta strands. So this is the jargon that we typically use in our field in terms of describing protein structures. But ultimately what these are in fact point clouds of atoms. Now, most of the work in our lab has been really trying to explore how to represent molecules differently. And this actually has been our representation of choice, which is molecular surfaces. And one of the things that you can do with these molecular surfaces is that, in fact, you can map on it, and you'll see how we're going to use this in the next slides. You can map onto it different types of features, and we can talk about geometrical features or chemical features. So this will come back next. And so the question that really drove what we wanted to ask was, so let's say that we have these molecules. Now we can describe them as protein surfaces. So can we actually identify surface patterns in these molecules that tell us about the biological function that these molecules carry in cells? And I like to do the analogy into when you start to learning features on people's faces, which can basically tell them where do they come from, how are they feeling in that day. So that's basically what we wanted to learn also about these molecules. And in fact, maybe most of you don't know, or maybe you do know, but actually working with continuous surfaces is a huge challenge in the field of protein modeling. And so we actually had to use a new suite of tools, which basically came from our long-term collaborator, Michael Bronstein, which is an expert in computer vision and geometric deep learning. And the first import of technology that we actually used was based on these geodesic convolutional neural networks that Michael uses to, or develops to analyze and learn patterns from 3D shapes. So the idea was to import basically this approach into the molecular world. And so in the way this looks is essentially this. So we created this software called Massive, which you'll hear this name a lot during this presentation, where we have a protein structure. We basically create a surface out of this protein structure, we segment it into geodesic patches, and then inside of these geodesic patches, you in fact have points that compose these surface areas. What we do afterwards is that we enrich these data structures with features, again, as I mentioned, geometric features and chemical features, which are, in essence, what determines the structure and function of proteins. And then using a couple of tricks from the geodesic convolutional networks, we can then actually create descriptors, which are no more, no less than numerical vectors, which can be trained for different tasks. And so this takes me to the first question of my talk, right, which is, can we actually read the functional fingerprints that are shown in these molecules, and can we use it for different types of applications? I will not go too much into the detail of each one of these applications, but the type of things that we were trying to predict were basically related. So if you have a protein structure, can you actually predict which ligand this protein structure is going to bind to? So you can imagine this is important to know what these proteins do in cells and which ligands maybe you would like to design in order to develop new drugs or things like that. Another aspect that are very common in protein bioinformatics is to try to understand which proteins actually interact with other proteins. So a lot of what proteins do in cells is just basically to bump into other proteins and basically take a decision: do I stick with this protein or do I not? And this, in fact, it sounds like a super trivial problem, but it is not. And so part of what we used here was to try to figure out which fingerprints were expressed in the surface of these proteins and see if we could use those to try to predict which other proteins could actually interact with those particular proteins of interest. So I'll just show you one more one result particularly related to this exercise that we called protein docking, which is again this idea of figuring out which pairs of proteins interact with one another. And so here we actually developed an architecture which was a Siamese network in this case, which could learn how to distinguish which proteins are supposed to interact with which and which ones are not. And in fact, we could do this with a very high accuracy. But something that arose actually from these studies wasn't necessarily the accuracy. When we actually compare these results to other state-of-the-art softwares, you see, so here, if you would be a perfect algorithm, you would have hit 100. So neither the state-of-the-art hits 100 or we hit 100. However, we hit an analogous result in a fraction of the time. And of course, that time does matter. And so several orders of magnitude that could be gained here in terms of speeding these calculations. And now, of course, this enables us to do many other things that I'll tell you in the future. So from there on, just giving you an update on how this is essentially evolving, we realized that a lot of our initial software framework was in fact pretty amateurish, let's put it this way, and it was really hacked. And so Fryer, which is actually also here in the meeting, is set up to develop a more end-to-end architecture where, in fact, we can drive all the information we need from atom point clouds without having to rely on external softwares to compute features and whatnot. So you can actually do now this from the beginning to the end. Okay, and then also a new player coming to place, and which is Helio, which is also here with us today. And basically, Helio started asking the question: can we actually use this surface fingerprints to start identifying potential new molecules to engage the surface sites in proteins? And so just to show you one of the initial results that I got, and this was almost literally our first attempt, where compared to other state-of-the-art softwares, we are not as good yet, particularly in accuracy, but we're definitely getting there, especially when we start considering larger candidates. So, and so just to give you some examples on how we can actually start rebuilding small molecules, just basically using the protein structure information. So for the last part of my talk, I'm going to focus on this aspect of writing functional fingerprints into protein surfaces. So what does this mean? So in a way, what we're trying to do here, And, and now I'm going to completely over-exaggerate. We're trying to play God over here. So as nature basically evolved a number of different proteins and gave you a couple of different sequences that make organisms fit, here we're trying to just say, well, maybe nature has evolved them for different purposes, but for my biotechnological purposes, I need to have the control over this design process. So clearly here, we're trying to aim for a generative stage of this algorithm where we want to create new sequences so that they can do things for us. And I'm going to give you examples on how we actually have done this. So now, the challenge here is the following. Let's say that you have one target protein that you want to bind to. So let's say the spike protein from SARS-CoV-2, and you really don't want to have it around, right? The challenge here is that you want to design other proteins in the computer so that they actually bind and neutralize their activity. And so in a lot of what we're going to talk about today, that I'm going to show you in the next slides, is based on this premise that when you bind to other proteins, maybe you can inhibit their activities that you're not interested. There are many other applications, but this is perhaps one of the most ideal ones. So now, maybe when I told you about this challenge, you're saying, well, that sounds super easy, right? You should be able to do this. 21st century, AI this, AI that. We should have solved all these problems. Well, guess what? We didn't yet. And there's many reasons why we didn't. And part of it is related to the fact that we actually don't really fully describe or understand the way molecules behave. And a lot of the calculations that we do in a computer are in fact approximations of the system. Some of the approximations are related to the fact that some of the scoring functions that we use, they don't really accurately capture the forces that govern the molecular behaviors. We most of the times also don't consider solvent, but it is true, right? Molecules do live in water, and we just sort of like compute everything in vacuum. And another point that it's also not present in many of the things, in many of the ways that we described our systems in the computer is dynamics. So molecules are not static entities. They have motion. Okay, so how did we actually, within all these limitations, how did we actually try to tackle this problem? So the way that we tried to do this was basically by, again, using the surface fingerprints, which we learn from protein surfaces, and basically by defining a site in a molecule that we want to target. And then given that we have a super fast docking algorithm, we can actually search for other potential candidates that bind to that site very quickly. So here we're actually talking about the order of 100 million fragments, so a lot of fragments. So once you actually do this search, what you do come out of this stage is that you have a protein target, again represented here in blue, and these are putative candidates. Once you have these candidates, then it starts the more experimental part also, where you transfer these motifs to other proteins, and then you go and you express them in the lab. So I know that many of you also don't think about how to make proteins in the lab in your daily basis, perhaps. So let me just tell you how exciting this is. So you go to your computer, you make a new sequence, and then you input that sequence into some web server, which basically connects to San Francisco on the other side of the ocean, and that codes into DNA. Those people over there synthesize the DNA that you're interested, codes for your protein. You then, when you receive it back here in Lausanne, you put it into a cell, and those cells basically start acting as the factories of your proteins, of the ones that you've designed in the computer. Then there's a number of steps in between. You purify your protein of interest, and then you go and test it. So that's basically how the process goes from the in silico generation into you actually are able to test something. Now, how do these things actually look just when we were in the computer? So the way that they looked is we started having motifs that look like this and motifs that look like that. We then transplant them to different proteins. And ultimately, we made these proteins, and we did one experiment, which I'm not going to explain you in super detail, but let's just say we can actually check if these proteins bind in vitro. And basically what you see here is essentially numbers reporting the strength of this binding interaction. And oftentimes what you are looking for is for proteins that really bind very tightly. But okay, but if you are a computational designer, and when you submit your papers to journals, the first question you get is your prediction right? And there's only one way to show if your prediction is right or not, is you have to go and solve a crystal structure, so real data, that you then compare to your model. And so that's what we did here. So basically you have a crystal structure which was solved in the lab, so the real structure that we have around, and the model that we had generated using all these massive fingerprints together with all the rest of the strategies that we have. And so in it, what you can see is that basically when the atoms of your model superimpose with a real structure, you call this a success, because in fact you predicted the binding mode of your newly designed protein. So we did this once, and then we also showed in a different case. And so to give you just one last example that perhaps I've been talking about it for the whole presentation, we also have done the same exercise, for instance, to design a binder that can engage the spike protein. And again, here you see how this binder looks like. So this is just a part of the spike protein. And then you test if it actually binds to it. And lastly, you actually test if it's able to neutralize the virus, which in this case, you'll see that it's able to neutralize the wild type virus, does not neutralize the Delta virus, but it neutralizes the Omicron virus. So you guys are safe for now. So that's just basically demonstrations of what we can use this technology for. And I'll just leave you with some of the conclusions, where essentially what we developed here was a new representation for protein structures. I don't think necessarily that this representation is limited to protein structures. We're exploring now different kinds of other molecules. However, what we ultimately generated was these vector fingerprints or embeddings, which were able to capture both geometric and chemical features of these molecules that will allow us to do multiple different things with it, including fast docking simulations, as well as generating new protein sequences that we can then validate in the lab. So there's, of course, many things to keep doing, and particularly on improving the architecture, working on protein ligand interactions, and start doing systems levels protein interaction network prediction. And with this, I thank you for your attention, because you guys really look like you cared, which is very good, and sometimes it doesn't happen. And also a word for the people that have worked in the lab. It really takes many different types of efforts to bring these molecules from life, from the computer to the wet lab. And I really have been very fortunate in working with a number of very smart people and motivated people. Thanks again for your attention.
We have time for a few questions.
Hi, my name is Adam. I'm really fascinated by this, and the reason why is because you say that we can understand protein molecule interactions just purely based on surface conformities between the two structures. My knowledge of proteins says that perhaps this is much more surprising than the way you have put it, because there are molecular interactions, potentially long range within the protein, that determine their interaction. So my question is, how much can you, for example, predict things like allostery in proteins, in enzymes, where maybe you have a binding site, an allosteric site, and they have some long-range interactions?
Okay, great question. You're clearly not a newbie on proteins, because if you know how to talk about allostery, that's not like the guy that just read a news on the 24 hours. So look, allostery has this thing that it requires motion. Here, we mostly taking care of these proteins in static snapshots. So we cannot predict any kind of allostery. I thought you also talked about something that is super interesting, which in a way it's perhaps one of the aspects which we still have a lot of difficulties, which is these long-range interactions, right? So typically when we talk about long-range interactions in terms of physics and chemistry, we're talking about electrostatics, right? And electrostatics for us is still something that remains complex, let's put it this way. And I think that right now we're certainly much better in short-range interactions than long-range interactions. And the reason for that is because a lot of what we learn is related to patterns of geometric complementarity. And geometric complementarity, by definition, is a short-range interaction, if you see what I mean.
Thank you. There's time for one more question.
Thank you for your talk. I was also wondering, because you look at the surface of the protein and also like what type of molecules is inside, would you also be interested to combine it with the graph structure that could like also embed information that is internal to the protein as well?
Yeah, so I think that's, there's perhaps two aspects of the description of molecules that I think it will enable us to do things that we can't do right now. So what you just described is one, is perhaps you could describe the backbone of the protein, which is basically the core, as a graph, and then try from there. Another aspect that we have not looked into much, and this was by design, but I think we need to change that, is in the sequence component. So, our representations are completely sequence agnostic. They have no idea. So if we actually would be able to include those two aspects, we could actually try to get to a unified representation of proteins where we capture much more of the features that determine their function than what we're doing right now. So yes, I think you're right.
Thank you.
Okay, thank you. I think there's clearly more scope for questions, but I think we have to move on in the agenda. Bruno, thank you very much.